Next I want to introduce Julie Orlando from CHOP. She's going to be talking about quantifying neuromotor behavior in STXBP1 individuals using wearables.
Hi, everybody. My name is Julie. I'm a physical therapist, and I have a PhD in biomechanics and movement science. And I'm really excited today to talk to you about quantifying neuromotor control using wearable sensors in individuals with STXBP1. I joined the ENDD team about 2 years ago and have been working on this project ever since. All right, so I'm clicking and nothing's happening. Got it, thanks. All right, so this figure is probably familiar to many of you in the room. This is from Julie Shen's paper, and it presents data from a retrospective cohort study. There were 534 individuals who showed impairments in neuromotor control. This included abnormality of movement, involuntary movements, ataxia, and tremor. And what I wanted to point out was that in that bottom right, all of those different areas all impact neuromotor control. So neuromotor control is part of the phenotype in STXBP1 related disorders. And this is data from the Citizens Health database. And what I'm pointing out on this slide is that we looked at different HPO terms related to movement disorders across several developmental epileptic encephalopathies. And abnormal movement is very common across multiple different of these DEEs. But ataxia occurs less frequently, and tremor primarily occurs in individuals with STXBP1. So tremor is a unique feature related to STXBP1. Also notable is that the frequency of tremor was higher in these real life citizen data and our STAR cohort than it was in the retrospective data set, approaching 68.5%. So there's very limited literature describing this tremor in STXBP1. There is a single study, and if the authors are here, I would love to meet them. But this was from Lucerne et al., and it was an EMG study which looked at 6 individuals with tremor. And the figure is from their paper, and it's from 2 different participants. Their arms were at rest or their arms were outstretched, and they did EMG in different muscles in the upper arm. And what I want you to notice is that there is this repeated 10 hertz frequency in the B figure, in the arms outstretched. And then there's sort of this rhythmic but irregular signal in participant 2. And the tremor was classified as being regular or irregular with large duration range with frequencies of 4 to 10 hertz. So that's what we know about tremor to date from 6 individuals. So we need to understand more. Anecdotally, families report to us that tremor is more obvious upon waking up and that it is brought out more during fine motor tasks. But we need ways to be able to measure this. But there are obstacles to measuring neuromotor control. How do I play the videos from here? Aha, okay, beautiful. So there are obstacles to measuring neuromotor control. Standardized assessments, such as the Bailey assessment, measure developmental skill and function, but they don't measure movement quality. So if I play that video one more time, hopefully. There we go. You can watch him put the peg inside the pegboard and then ask for more, which is so cute. But you can see that it's challenging for him. So he would have scored a 1 on the Bailey assessment, because a 1 out of 2 points, because he was able to get 2 of the pegs in the pegboard, but he wasn't able to get all 6. But we don't know anything about the quality of that movement from that score. Movement quality assessments also tend to require multi-step directions. So there are scales for ataxia, but these are really hard to do within this population. So sensors have the potential to be used to measure neuromotor control and the benefit of potentially being used within the clinical environment or in the home setting. And they've been used to measure movement quality in other populations. In Parkinsonism and essential tremor, you can actually use sensors to distinguish between the two types of tremor when you put the sensors on their upper limb, and then individuals perform some daily activities and then drew these Archimedes spirals. They've also been helpful to identify tremor, they've also been used to help identify Friedreich's ataxia. The signals from the upper limb were related to posture and locomotion differences in individuals with Friedreich's ataxia. They've been used to predict autism spectrum disorders in infants who had a family member who was high risk. And they've been used in spinal cerebellar ataxia. They were actually more responsive to gait changes than other clinical measures. So I hope I've set the stage that we should be using sensors. And now I'm going to talk about our study where we quantified neuromotor control in STXBP1 related disorders compared to typical development and evaluated the relations with existing developmental assessments. I'm sorry, this figure is coming up a little bit blurry, but we'll just keep moving on. But these are the methods that we used. And so we had individuals, we placed sensors on their upper limbs. We completed a structured reaching task where they reached for items. And then we took the data from each of those 4 sensors. They were uploaded and processed in MATLAB. I did some signal processing where we took the acceleration data and the XYZ axes and the gyroscope data and the XYZ axes. And then we, when that was done, we took the resultant acceleration. And so we ended up with sensor data from 4 limbs. So we have a dominant upper arm and forearm sensors, and then non-dominant upper arm and forearm sensors. So with the forearm sensors, with the dominant arm sensors, we did between group comparisons between the typically developing controls and individuals with STXBP1. And then with all the sensors, we did tremor prediction using a machine learning model. So the first thing we found is that evaluating tremor with wearable sensors was feasible in the clinical setting. This is not a given. If anyone has attended any of our clinical days at CHOP or at the other sites, you know that they're very long days. It's a busy environment. The rooms are small. It's a little bit chaotic. We try to control the chaos. But it was feasible. We were able to collect data for 31 individuals. 5 individuals were not able to reach, and 6 individuals reached with only 1 limb, but their data was able to be included in this analysis. We called that their dominant limb, as it was their reaching arm. And then there was technology failure with 5 individuals, which was usually just a battery died, more human error than technology failure. And then our typically developing sample, there were 19 individuals who completed the reaching assessment, and 1 did not reach, just because sometimes kids don't do things that you want them to do. So I want you to look at these videos and think about what you want to measure. So here's our first child with STXBP1. She's doing a reach. You can see the tremor when she's doing that reach, and you can see it when she's playing. She's going to reach again. So you see the tremor come out. And now I'm going to play the video of our typically developing control. This is my niece. No family parties are safe from my data collections. And so there's obvious differences between the two that you can see with your eyes, right? So now we have to decide, what do we want to measure? How are we going to try to capture these differences that we can see? So the first thing you see is the duration difference. And then there are differences in intensity, just the magnitude of the movement. There's also differences in the smoothness of the movement. And then there are frequency-related differences that we're going to explore. Okay, so I'm going to now describe how we calculated these different metrics, just to get everybody on the same page. You're all going to become biomechanists for a few minutes, but bear with me. So So what these two figures are showing is our individual with STXBP1, which is on the left, on your right, sorry, and the individual with typically developing. And so the one that is a lot more wiggly is our STXer. So these are time series plots of acceleration magnitude comparing just the forearm sensor and the dominant limb. And what I want to point out first is the duration difference. So on the x-axis, there's that diamond shape, and that shows the duration difference between our STXer and typically developing control. These are 3 reaches that are put together for both individuals. So because one reach is just too difficult, it's too messy just to do one reach. So we need to have a variable that's a little more robust to slight differences. So we picked 3. We got those 3 by finding the median reach duration and picking the 3 reaches that are closest to that median. That gives us some flexibility because, as we said, kids don't always do what you want them to do. So we measure reach duration. We also measure the measures of intensity, which are this root mean squared acceleration. That's that red line that you see dotted. We measure the peak acceleration, so that's the upper peak of the acceleration signal. And then we measure the acceleration time area, and that's the area under the acceleration curve. And then we measure the acceleration peak count. So that's how many times that spike wave goes up and down. And that's actually a measure of smoothness. The other measure of smoothness that we do is called spectral arc length. So this is the normalized power spectrum of the data. So now we're looking in the frequency domain, and we're taking the negative sum of all of the arc lengths along that signal. What I want to point out is that in our typically developing sample, the y-axis, you see this kind of upper peak. I'm sorry, I want to go back one slide and show you that. So I want to point out that the acceleration signal on the y-axis for our typically developing individual is higher. So that goes up to 15, whereas in our STXer, the magnitude only goes up to 6. So we're going to start there. Okay, now I've set the stage. Now we can talk about smoothness. And so in our typically developing control, there's this high peak and then kind of a gradual decline, whereas in our STXer, there's a more narrow peak, and then there's a little more wiggliness in the bottom. The wiggliness that you're seeing there is the smoothness measure. Okay, and then there are frequency-related measures. And so these are other measures in the frequency domain. So we can look at the high frequency band power, and that is yeah, you can see the shading, great. Sorry, I cannot see it. So there's shading, that the high frequency band power is that shaded area. So individuals move, usually voluntary motion occurs between 0 and 3 hertz. So that's your usual activity. Anything that happens above 3 hertz is typically a tremor-like movement. So that's how we can sort of gather where tremor is happening in this frequency domain. So we took the high frequency band power of anything above 3 hertz, and we took the area under that curve. And then we looked at the peak frequency within that high frequency band. So for our typically developing individuals, that's pretty much always right at 3, because that's, as you see that great gradual decline, it's sort of arbitrarily set there because that's where we've established the high frequency band. And then we took the full width half maximum, which is also a measure of smoothness, and it's getting at like the narrowness of the frequency. So if someone had a tremor that occurred at one specific frequency, we would see a peak right at that frequency level. Anything that is a more oscillatory is going to be a more broadband signal. And then we took the mean tremor power band also, which is looking at, that's that like dashed line. Looking at the mean band. So now you guys have an idea of what these measures are, and now I can show you what happened with them. I'm happy to dive more into the biomechanics if anybody wants more information, but I probably have put you all to sleep in the last 3 minutes. So I'm going to move on now to our plots that are much more exciting to look at. So this is a lot of data on one slide, but I want to draw your attention to just the fact that there are a lot of differences between our typically developing group and our STXers. across all of these different measures, tremor is denoted in orange. So not all individuals with STX have tremor, but some do, they're in orange. The only metric that wasn't significantly different between the two was acceleration time area. But because we don't know anything about these metrics, really, nobody has done this before in this population or put all these things together, I wanted to know more about what the distribution looked like within these metrics. And so we took the z-score. So these are density plots of the z-score, showing the distribution between the groups, trying to see how the typically developing stratified from STXers. And you can see visually that many of the metrics look different between the two, but some have some overlap. And then we looked more at the individual level in that bar plot, which is showing just within our STX cohort how many were 1 standard deviation, 1 to 2 standard deviations, 2 to 3, or more than 3. How many of those metrics were different? So how many individuals had 1, 2, or 3 metrics that were within or outside those standard deviations. And so most of the participants had metrics that were more than 3 standard deviations, but there also had some metrics that were within the 2 to 3 range or 1 to 2 range. So there's a lot of variability across the different metrics. So we haven't found like one signal that we can say, this is it. This one metric is going to be always different between our STXers and our typically developing controls. But overall, when you look at this together, there are differences between these different metrics that we've evaluated. And there were relations between these metrics and development, but not everything was correlated, which again points to that movement is different. Motor control is different than developmental assessment movement. We then put their different metrics into a machine learning algorithm, and this is showing all different types of sensor configurations. And so what I'm trying to draw your attention to in that little purple box is that the area under the curve for all of our different sensor configurations was between 0.72 and 0.84. So they're all pretty high. So you can use any combination of sensors, but the best one was the red one, and that was using bilateral upper and forearm sensors. So that was all of our sensors combined, gave us the best result at predicting tremor. There were specific features that were most predictive of tremor, and they tended to be the signals that were in the frequency domain. And so I'm just going to kind of start wrapping up here. We're almost done. Wearable sensor metrics are promising to evaluate motor control, including tremor, in individuals with STXBP1 related disorders. Sensor-based assessments were well tolerated across all ages and developmental abilities. Motor control metrics were able to be distinguished from our STXers and our typically developing controls, and a subset of these sensor-derived motor control metrics correlated with developmental outcomes, but they did not correlate perfectly, which again points to that function and motor control are not the same. And motor control metrics were predictive of tremor using supervised machine learning. In development is automating our pipeline. So we are working on automating our reach detection using video, and that is coming along. And next steps are working on evaluating more fine motor tasks with increasing challenge, which I hinted at today from the one video where you saw the individual with all the sensors on doing that Bailey item task. So I have some references and some acknowledgements. Thank you very much to the team, and I'm happy to take some questions.
Time for 1 or 2 quick questions.
Great talk. So in some of our neurologic assessments, we specifically tried to pay attention to tremor and look at some items for it as part of our severity assessment. And it didn't work out great. And we were trying to figure out why. We kind of hypothesized because not everybody has it. So some patients do, some don't. It looks like you actually had a higher proportion of patients who had tremor than maybe we found in the like broader cohort. And I'm just trying to distinguish, you have a sensitivity advantage here with this method. We tried to quantify also how much does this seem to be impacting function, but that's a bit of a gestalt of our like exam. and we're trying to subtract out their baseline developmental status, right? So like accounting for that, how much is it additionally contributing? I'm wondering if how you've tried to sort of piece those components out to see what the relative advantage of this method is.
Yeah, these are all the things that keep me up at night. But I absolutely agree. These are all really important points. So I think we're not great at evaluating tremor in the setting of a neurology appointment. I say that with all love to our neurologists, but I think as we've seen parents report that the tremor kind of varies throughout the day. It's worse when they wake up. It's worse when they're doing tasks. So if you're not having them do that task, you might not see it. There have been times when, I've looked at Dr. Helbig's note and be like, hey, actually, I did see tremor when he did this. Dr. Helbig did a beautiful job of his developmental, his neurology assessment, but he didn't have him do the Bailey, like he's not in the room when we do the Bailey. So sometimes you see things when you do specific tasks. So that's what I'm hoping our next step of measuring those fine motor tasks is going to capture. So it's a long answer to a short question, but I think there's a lot more work to do, and I think we can work harder at standardizing some of our own assessments in the qualitative ways, and then also we can work to find these objective measures to get out those little pieces.
Clearly superior to Ingo.
You said that. I didn't say that. I like my job, Scott. Good.
Hi, this is Hannah Stemmiger from University of Antwerp. Thanks, it was a really nice talk. We actually did quite some research on tremor in adults with STXBP1 also, and what we saw that there's different types of tremor, like cerebellar, intentional, or resting tremor in some cases. So I wondered, is what you're doing actually helping classifying tremors and consequently treating them also?
Yeah, so I would love to do that as a next step. So at this point, we have now collected much more data than I've been able to process and analyze. But I do hope with a larger data set that we'll be able to get at some of those differences. I think the video is also going to help us with that because we'll be able to relate it to the differences that we see. But I do think there are potentially differences, and I'm hoping we can do some cluster analysis in the future to try to figure it out. And then, yeah, the goal is always treatment in the end, but that's somebody else's job. Thank you.